Hello class. So in our we were doing chapter 6, that is seed, the structure and germination. I hope you have already viewed a video which I posted last time regarding the structure of a seed in detail. If you have not gone through that, please go through that in order to understand the next topic related to this chapter, that is germination. So today we will be carrying forward with germination. Now first let us understand what is germination. Okay, so germination means whenever we all know that inside the seed there is a small baby plant, also referred to as embryo. So when this embryo gives rise to seedling, that process is known as germination, and this seedling only is later on going to give rise to a new plant. Okay, inside every seed there is an embryo. That embryo later under a favorable condition, okay, it's going to give rise to seedling. That process is known as germination. Okay, in a dry seed, this embryo is in a dormant stage. That means it's in a resting phase. From outside it looks like as if nothing is going on inside, but inside the seed different chemical reaction is being carried forward, and whenever it gets a necessary condition which is favorable for it, that particular seed is going to break open and your embryo is going to get converted into the seedling. So okay, let us understand the conditions which are necessary for germination. Okay, the first one would be your water. Water is required for germination. Now what is the purpose of water in regard to germination? Number one, your water, it seeps through the minute hole that is present in the seed. That hole is referred to as a microfile. That is also the reason where your pollen tubes comes and fuses with the ovule, right? We have already discussed during fertilization, while studying about fertilization. So through that minute hole called microfile, your water is going to seep inside. Okay, and we all know that seed is protected by outer tough covering called seed coat. So whenever water seeps inside, the water is going to swell upon. That process through which the water seeps inside is called imbibition, and that seed is going to swell upon, and your seed coat will be rupturing. Okay, whenever the seed coat ruptures, your radicle will elongate and ground itself and later on form a root system, and your primule is going to come out and later on form a shoot system. Okay, so it's going to give rise to a radicle. So first, why do we require water? For rupturing the tough seed coat. Okay, number two is, what is the other requirement of a water? Since different chemical reactions are going on within the seed, so for carrying out those chemical reaction, water is very essential. And also for enzymes to act upon the food that is being stored in a cotyledon or in endosperm, right? Food is stored in cotyledon or in endosperm. So for enzymes to act upon that food, so that food can be broken down into a simpler form, so that food can be taken up by the embryo, just like in our body, right? Whatever the complex food we eat has to be first broken down into a simpler form, that is glucose. Only then it can be absorbed by our body. Similarly, even within the seed, the food is stored in a complex form, so that complex food has to be converted into a diffusible form by the enzymes, and for the action of the enzymes you need water. Okay, so those two were the important uses of water for germination. Now next one is your temperature. Now too low temperature, the optimum temperature for germination is around 25 to 35 degrees Celsius. Lower or higher temperature would not favor germination process. Okay, now so why it would not favor a germination process? Because in a higher temperature your enzymes would not be working. Okay, your enzymes, for enzymes to work, it also has an optimum temperature. So your enzymes which are essential for breaking down of the food or carrying out other different chemical reaction may get denatured under a higher temperature. Okay, because and under a lower temperature it might not work. So your temperature also has to be optimum, that is between 25 degrees Celsius to 35 degrees Celsius for germination to occur. Okay, now next one is your oxygen. Why do we need oxygen? we need oxygen in order to carry out respiration, right? So similarly, inside the seed also, since cell division is occurring, cell is dividing in order to form a seedling, okay, so continuous cell division is taking place. So for those cell division, energy has to be provided, right? So for providing energy, what has to be done? Your food has to be broken down, right? So that means respiration process has to take place. So when does the respiration process take place? Respiration process takes place only in the presence of oxygen, right? Aerobic respiration. So for breaking down of the food, in order to provide the energy to carry on with the cell division process that's taking place within the seed, you need oxygen. Okay, so these are the three essential conditions which has to be met in order for germination to occur. Okay, even if one of the condition is not met, germination is not going to occur. So next we will be following up with the experiment which is going to prove that these conditions are essential for germination to occur. Okay. Okay class, now let's carry on with the simple experiment which is going to prove that water, oxygen and your temperature is very much essential for germination to takes place. This experiment you can carry out at your home also. You can these are very simple experiment, so you can carry out this experiment at your home. So first let us carry on with the experiment which will prove that your water is necessary for germination. Okay, now let us look at the experiment setup. Now let us look at beaker A. So what do you have? You have two beakers. Okay, in both one of the beaker, if you look at beaker A, you just have a dry cotton, okay, and on top of the dry cotton you have just placed a seed. Okay, now look at beaker B. You have a wet cotton there, that means your cotton was soaked in the water and then placed there, so you have a wet cotton there. Okay, and you have placed a dry seed in both the cases. Okay, now what can you visualize after a few days? That in your beaker B, seedling starts appearing, that means your germinated seed, you can see the germinated seed in beaker B. Okay, but in case of beaker A, no germination of the seed occurs. Okay, so what does this prove? This proves that water is very much essential for germination to take place, because in beaker A you did not put water, you did not place water on the seed. Okay, but in beaker B you had water. Okay, so since you did not have water in beaker A, so germination did not take place. So for germination process to take place, you need to have water is very much essential. Okay, now next experiment is to prove that temperature is essential or necessary for germination. Like I told you, it has optimum temperature of 25 degrees Celsius to 35 degrees Celsius is very much necessary for germination to take place. Okay, now let us look at the experimental setup. Okay, what do you do is, you in this case also you take two beakers. Okay, beaker A and beaker B. You take the wet cotton in both the beakers. Okay, and then you place a dry seed in both the beaker. But beaker A, you place it inside a ice cube, or you can place it in the refrigerator at your home. Okay, temperature in refrigerator is usually 0 degrees Celsius to 7 degrees Celsius. Okay, lower temperature like 3, 4, 7 degrees Celsius. So you have very low temperature in refrigerator, whereas beaker B you keep it at a room temperature. Okay, so what can you visualize after a few days? That if you whatever the beaker A which you had placed in a ice cube or refrigerator, what can you see? That no germination has occurred. Okay, but in case of beaker B, the seed had germinated. So what does this prove? This proves that temperature is essential for germination. Okay, and your temperature has to be around 25 degrees Celsius to 35 degrees Celsius for most of the seeds. okay, but there are exceptions. So for most of the seed, the optimum temperature is between 25 to 35 degrees Celsius, okay. Lower or higher is not gonna favor germination process. Now next experiment is to prove that oxygen is essential for germination. I told you, right, stored food has the seed need energy. Now for energy, providing energy, the food has to be broken down, and that food is broken down only in the presence of oxygen. So what would you so oxygen is required by the seed for germination. So in order to prove that germination requires oxygen, what will you do is, you see the experiment setup. Flask A and flask B, both of them have a wet cotton, or your grams are soaked in a water, okay, your beans are soaked in a water, and in a big flask A, in a small test tube, can you see the small test tube here? In a small test tube you have put a pyrogallic acid. Now what is the function of this pyrogallic acid? It absorbs whatever the oxygen, if it is present within the flask, okay. And in flask B, you simply place the test tube with water, okay. Now what will you see is, and you have put a stopper on both the flask A and flask B. Why have you done that? So that no oxygen from outside can enter the flask, okay, in order to avoid the error, because if you do not place a stopper, then oxygen from outside will enter, and again germination is going to take place, right? So in order to avoid that error, you have placed a stopper to prevent the entry of oxygen from outside, okay. Now what will you see is, after few days, in flask A, where your pyrogallic acid was present, okay, no germination takes place. Why? Because whatever the oxygen was present inside the flask has been utilized by the pyrogallic acid, right? So no breakdown of the food, no energy being released, that's why your germination does not take place here, okay. But in case of flask B, where you had placed just the water, there you can see that germination has occurred, okay. So what does this experiment prove? Now this experiment proves that oxygen is very much essential for germination, okay. Now next is your 3 bean seed experiment. Here we will be taking all these 3 factors, that is oxygen, water and temperature, normal temperature, and we will be carrying on the experiment. What will you do is, first, in a slide, you all have seen slide, right? You will take 3 seeds, you will place 2 seed on the edges of the slide, okay, and 1 in the middle portion, okay. You will tie it with the help of a thread. Then you will take a beaker half filled with water, and then you will place a slide which had a seed on it. Now you will place a slide in such a manner that one of the seed is completely submerged in water, okay. The second one is partially submerged in water, and third one is in the air, that means it is not getting any water, okay. Now what will we, and we'll keep this setup for few days. After few days, what will we see is, in the seed which was completely submerged in water, no germination took place because it did not get the sufficient oxygen, which is also essential for germination, okay. Now in seed, the second seed which was partially submerged in water, germination took place because it got the water which was needed for germination and even the oxygen, okay, which was needed for germination. So germination occurred in second one. But if you look at the third seed, which was in the air, now this seed got the oxygen, temperature was normal, but no water. That's why no germination took place in this particular seed also, okay. So these were the experiments to prove that for germination to occur, it requires water, optimum temperature and oxygen, okay. In our next class, we will be discussing about the types of germination. So I request you people to go through the videos that we are uploading, okay, so that it becomes easier for you to understand whatever we'll be teaching next. Okay, thank you.